Hi, I'm Priya Shah, and I'm an assistant professor here at UC Davis in the departments of chemical engineering and microbiology and molecular genetics. I'm here today with some of my colleagues, Karen McDonald, Roland Fowler, and Soman Nandi, who are also professors in chemical engineering. We would like to chat with you about some of our work on creating important proteins that will be needed to fight the COVID-19 pandemic. Specifically, our goal is to produce viral proteins quickly and in large amounts for tests to determine if someone is immune to COVID-19. This is important because the COVID-19 pandemic will require the production of an enormous amount of proteins in a short period of time for these tests. And ramping up production of these proteins at the industrial scale can be laborious. And we're using experimental and computational approaches to cut down on production time and improve yields of high quality proteins. We're focusing on producing a SARS coronavirus 2 protein called spike. Spike is what we call a glycoprotein, or a protein with sugars attached to it. And this is a protein that's a major part of the viral particles that make up the virus. And this is also what our antibodies recognize to provide us with immunity. And so producing a large amount of these proteins will be critical to reopening the economy, since it's a major component of the tests that will determine whether someone has immunity to SARS coronavirus 2.
CHO and other mammalian systems potentially can produce these high quality target proteins. These systems are expensive and slower and to scale up due to the several factors. On the other hand, bacterial and insect systems can also produce the same molecule, although the differences in sugar that Karen and Roland will talk about it and the protein folding have resulted in lower efficacy produced in these systems compared to the mammalian or CHO cells. On top of everything, whether it is therapeutic, vaccine, or diagnostics, the predicted quantity needed will be remarkably high in order to protect the entire global population. We know worldwide several companies and researchers are working hard to resolve this issue, but our preliminary calculation shows there is no way existing manufacturing facilities can meet this global demand, particularly in quality required and within affordable cost in near future. To balance this production time and cost with protein quality, we will also test our protein produced in Nicotiana benthamiana, a relative actually of tobacco plants. The production advantage is speed. It takes about only a week to produce, high production level, low upstream production cost, and linear scalability. Essentially, here we are using plant as a single-use disposable bioreactor.
One particular aspect we need to study in this project is what is called glycosylation. Proteins like the spike protein have sugars connected to them, and these sugars vary even for the exact same protein. This is one of the reasons why it is hard to determine how proteins interact. Particularly if you make the exact same protein using different cells, like plant cells or mammalian cells, we will get different sugars. Also, we will study if we can use smaller pieces of the protein without compromising accuracy, which would simplify production at large scales. Computer simulations are a very useful complement to experiments. As in experiments, we cannot really see the atoms, except in very specific circumstances. In simulations, we know what all the atoms are doing, but the simulations rely on the quality of the model. We have to validate the simulations against experimental data. Such collaborations are clear examples where the total is much more than the sum of its parts.
This collaborative research project is really focused on practical engineering issues related to producing these important diagnostic proteins, improving production efficiency, ensuring product quality, all the while keeping cost effectiveness and the ability to quickly scale up biomanufacturing at the forefront. As Soman has described, using plants to make these important diagnostic proteins is a promising approach, one in which UC Davis is uniquely positioned because of our campus strengths in plant sciences, biotechnology, genomics, bioanalytics, and bioprocess engineering. Similarly, Priya's work with mammalian cells builds on research expertise from the School of Medicine, Veterinary Medicine, and Engineering. We can also utilize the many unique capabilities that UC Davis has. Laboratories that can work with the SARS-CoV-2 clinical samples and do testing with our proteins, animal models ranging from mice to non-human primates, and state-of-the-art core facilities to help us characterize our proteins. Roland, do you want to add your thoughts?
UC Davis, as the most comprehensive University of California campus, really makes this campus a natural choice for such projects. One of the biggest strengths of UC Davis is actually the collaborative atmosphere. We have such a wide variety of expertise, particularly in the life sciences, and everyone is interested in collaborations.
Thank you, Roland, and thank you everyone for explaining how chemical and biochemical engineers have a big role to play in fighting the COVID-19 pandemic. Our research will hopefully make the rollout of mass-scale testing more affordable and timely. And finally, I think one of the most important things to take away from our research is that the approaches we're developing here for COVID-19 are actually broadly applicable to many different scenarios where time is of the essence. This could be equally useful for another pandemic in the future, for bioterrorism threats, or even producing medicines on demand for the first people to travel to Mars. We're just thrilled to be able to contribute to the COVID-19 response, and we're grateful to the UC Davis Office of Research for funding our work. Thank you.